welcome to the session. We will see a case scenario first. A group of surgical site infection cases have occurred in a neurosurgery ward, and the culprit organism was Acinetobacter species. And it was noticed that all the cases were operated in neurosurgery operating room. Okay. So after that, an outbreak investigation was carried out. So what type of outbreak investigation you have to perform? So it's a case of surgical site infection due to Acinetobacter species, and all these cases were operated in neurosurgery operating room. Okay. So you have to conduct environmental surveillance. Okay, where you have to see that whether the air microbiology and surface microbiology of the operating room, you have to study properly whether there is a contamination of the air or the surfaces, you have to examine thoroughly. Okay, so today's session is on environmental surveillance. Environmental surveillance plays a very important role in hospital infection control because you have to see that the various environmental objects such as water, air and surface of the hospital are free of organism or not, because you know that environment plays a very important source of infection. We have discussed that the source of infection in a hospital to cause various hospital acquired infection, the source of infection, it could be from the healthcare worker, especially the hands of healthcare worker, or the environmental sources, or it could be other patients. So these are the three sources from which the infection may spread to the patient in the hospitals. So environment is one among them. So you have to perform environmental surveillance such as water surveillance, air surveillance and surface surveillance from time to time, as and when it is needed to rule out any environmental contamination of infection. So we'll start with the water surveillance. So we will see what are the indications of environmental surveillance. CDC has given guideline. They say that the microbiological sampling of air, water and surfaces, that is environmental sampling, is an expensive and time consuming process. Therefore, regular or routine environmental surveillance is not indicated. Okay, random periodic sampling is not recommended. Only when the specific indications are present, you have to conduct environmental surveillance. Okay, if somebody will ask you that regularly every month whether you have to conduct air surveillance or surface surveillance in the operation theatre, answer is no. Random periodic sampling is not recommended. You have to do something called as targeted microbiological sampling, which involves when you have an outbreak investigation. The case scenario what we have discussed, you have an outbreak of surgical site infection. This is the most important indication of conducting environmental surveillance to evaluate the change in infection control practices. Suppose you have introduced a new infection control practices, to know that whether the newly introduced infection control practices is working out properly or not, you may have to conduct environmental surveillance. If any construction is going on or following the renovation activity of the ICUs and OT, you have to conduct environmental surveillance. And last but not the least, for any research activities also. So these are the various indications of environmental surveillance. So there are three things in the environment: air, water and the surfaces. So we'll start first with water surveillance. Hospital water and the various water containing devices of the hospital, they may serve as a reservoir of various waterborne pathogens in the hospital. Okay, so microbiological contamination of water in healthcare setting, it may be classified in two ways. Category 1 pathogens are the enteric pathogen. Enteric pathogens, the classical example, which all are the pathogens that are transmitted through water and will cause various enteric infection. The classical example is Shigella, Salmonella, okay, Vibrio. These are the various enteric pathogens which are transmitted by enteric route, and they are transmitted through the contaminated water. So this is called as category 1 pathogen, enteric pathogen. So this is not only important in healthcare setting, it is not only important in hospitals, but also it is important in community. Drinking water should be free from enteric pathogen. This is what you have to ensure, that the drinking water, whether it is hospital drinking water or community drinking water, it has to be free from the enteric pathogen. Another category is called as common hospital pathogen. These are the various hospital multi-drug resistant organisms of the hospital. The classical example is Pseudomonas, Acinetobacter, okay, all this, and Escherichia coli, Klebsiella. So there are various hospital acquired multi-drug resistant organisms. These are exclusively present in the hospital environment. So it may happen that the hospital water supply may be contaminated with this MDROs. I would like to say that there are two kind of water as far as hospital is concerned. In hospital you have a drinking water. In hospital you have a water that is used for patient care activity, such as operation theater or maybe ICUs. So the water may also be used for patient care activity. So remember that drinking water must be free from category 1 pathogen. Okay, category 2 pathogens are the common hospital environmental contaminant, like common hospital environmental pathogens like Pseudomonas or Acinetobacter. These pathogens, if it is contaminated with drinking water, it is not a big problem, because they say that the drinking water should not have enteric pathogen, because it is an enteric pathogen. If it is contaminated with the drinking water, that will cause various enteric infections. However, common hospital pathogens like Pseudomonas, Acinetobacter, that contamination with the drinking water should not be a big problem unless otherwise it exceeds a particular limit. Whereas the water used in patient care activity, that is either in the operation or maybe in other patient care activities, that must be free from, not only from the enteric pathogen, but also from the category 2 pathogen, that is common hospital pathogen. So please remember, hospital environment may have two type of water. A drinking water is same like a community drinking water. So whether in hospital or in community, the drinking water must be free from category 1 pathogen, that is enteric pathogens, whereas water used in patient care activity must be devoid of any organism. Both category 1 and 2 organisms should not be present in the water which is used for patient care activity. So now we will discuss one by one. First we will discuss how we can test the drinking water quality. Okay, so please remember that the enteric pathogens are present in drinking water only in minute quantity. That is Shigella, Salmonella, Vibrio. These are the classical enteric pathogens. They are present in drinking water only in very minute quantity. Therefore, the routine bacteriological examination of water, it is impracticable. It is impracticable to attempt directly to detect the presence of enteric pathogen by routine bacteriological examination, because I told you that the Shigella, Salmonella, Vibrio, these are the routine enteric pathogen. They are present in very minute quantity. Therefore, testing for drinking water can be performed by detecting various indicator organisms. The indicator organism, the classical example is Escherichia coli. Fecal Escherichia coli is a classical example of indicator organism. The presence of these indicator organism indicate that there is a fecal contamination of drinking water which has been occurred. Okay, so how the drinking water may get contaminated? Drinking water, the most common source of getting contaminated is by contaminated with the sewage system. Okay, the fecal contamination of the drinking water is most common, and when you have fecal contamination, the various enteric pathogens which may be present in the feces that may get contaminated, as well as there are various fecal commensal that also may get contaminated with the drinking water. Because enteric pathogen in drinking water, the number is very less, minute quantity, therefore it is impracticable to isolate this organism in water. Therefore we indirectly detect the indicator organism. These indicator organisms are various fecal commensals which may get contaminated and which may be present in the water. The classical example is fecal E. coli, Escherichia coli, and they give an indirect evidence that the fecal contamination has taken place. I repeat, they give an indirect evidence that the fecal contamination has taken place. So, if fecal contamination has taken place, there is a chance that enteric pathogens would have contaminated with the water. So, that is how indicator organisms will give a indirect evidence that the enteric pathogens would have contaminated the water. Okay, so now we will see what are the property of indicator organism, because human GI has got various organisms. Commensals are enormous. Numerous commensals are there. So which are the commensals that are used as indicator organism? The important property of indicator organism include they should present in excess number than the enteric pathogen in water. Okay, the indicator organism should present in excess number in water. They should not be able to proliferate in water to any extent, so that whatever amount of organism you will detect in water, that will give a direct estimate that that much of organisms were present in the fecal sample also. So this is this will give you a indirect evidence that what is the quantity of fecal contamination that has taken place with water. Okay, so that is why they should not proliferate further in the water, so that you will not be able to differentiate that the quantity is because of proliferation in water, or it is actual quantity which has come from fecal contamination. Okay, the third property is they should show more resistance to the than the pathogens to the stresses of aquatic environment and disinfection processes, because the drinking water is usually treated with chlorine for disinfection. Therefore the indicator organisms should be more resistant to the stresses of the aquatic environment and the chlorination of the water. That is how they should be able to survive in presence of chlorination, and so that you can be detecting this organism. So now I will show you the list of various indicator organisms and their interpretation if they are found in drinking water. Okay, so these are the various indicator organisms. The most common indicator organism is coliform organisms. So coliform organisms, that is enteric coliform other than E. coli, that is Klebsiella or Proteus. All these coliform organism presence in water sample, it indicates remote contamination. Remember, it indicates remote contamination, and this is not a very important thing, because remote contamination is what the water sample has been contaminated with the feces long back. Remember that the enteric pathogens such as Shigella, Salmonella and Vibrio, they usually die in short time after water contamination. So after the fecal contamination of the drinking water, the enteric pathogen will not last long. They will die in a shorter interval of time. Therefore, remote contamination of the water is not very important, because it will not say that the enteric pathogens are present, because enteric pathogens will usually die off in a shorter period of time. Therefore, detection of coliform other than Escherichia coli indicate remote contamination, and if you have a remote contamination, it is not a very important factor, because it cannot confirm fecal contamination due to enteric pathogen. However, the next important indicator organism is Escherichia coli, that is fecal thermotolerant Escherichia coli. This indicate recent fecal contamination, because even a fecal E. coli also will die off in some time, and therefore fecal E. coli cannot be present after a long time of water contamination with the feces. Therefore, if you detect fecal E. coli in the drinking water, it indicates recent fecal contamination of water. Therefore, this is considered as the most reliable, most sensitive indicator of water contamination of feces. Okay, fecal contamination of water, there are so many organisms, but the most reliable organism is fecal thermotolerant E. coli. Okay, the next indicator organism include fecal streptococci and fecal clostridium perfringens. So these also may be present in the commensal in the feces. So they will confirm again remote contamination. They will not be able to say anything about the recent contamination. Okay, Pseudomonas aeruginosa is a least reliable indicator. It is useful in hospital water supply, because the detection of Pseudomonas aeruginosa in hospital water is very important, because hospital water is not only used for drinking purpose, it is also used for various patient care activities. So hospital water supply should be devoid of all organisms including Pseudomonas aeruginosa. Now coming to bacteriophages which are specific for Escherichia coli. Bacteriophage means what? These are the viruses that can infect the bacteria. Okay, so the bacteriophages which are specific for E. coli, they may indicate a fecal pollution of water, and indirectly they may also indicate viral pollution. Okay, so bacteriophages are used for fecal contamination of water with the respective bacteria in which they reside, and also they may indirectly indicate viral pollution of water. So these are the various indicator organisms, but among these the most reliable indicator organism is fecal or thermotolerant E. coli. Now we will see what are the testing methods available for water analysis. So you can see here there are three testing methods available: multiple tube method, membrane filtration method, and presence absence method. These are the three methods available for testing of drinking water. Coming to multiple tube method, it is extensively used for, it is the most common method used for drinking water testing, and it is usually indicated for highly turbid drinking water where the other methods you cannot perform. Membrane filtration method is less commonly preferred, and it is here the indications are very specific. It is useful for dialysis water. It is also useful for testing large volume of water, and it is also useful for testing clean water where the bacterial count is expected to be low. Okay, where the bacterial count is expected to be high, that is turbid water, the best method is multiple tube method. But by and large, membrane filtration method is more sensitive, and it is an expensive method, therefore it is not done for all water specimen. The use of membrane filtration method is in hospital setting is usually restricted to dialysis water and clean water where the organism load may be low, so a multiple tube method may fail to detect. In such case also a membrane filtration method can be done, and testing for large volume of water, membrane filtration method is the method of choice. Presence absence method is the least preferred method. It is not a reliable method at all. It usually monitors the quality of drinking water where the contamination is likely to be rare. Okay, it is not a very common method to be followed. Before we discuss the individual methods, let us discuss how you will collect the water specimen. Okay, collection and transport of the water specimen is very important. They say that at least 150 to 200 ml of water you have to collect, and after you collect the water you can neutralize with sodium thiosulfate. Sodium thiosulfate can neutralize the residual chlorine if it is present in the water sample, because you know that chlorination is done for the water sample for the disinfection, and presence of chlorine residues in water may inhibit the organism in water to grow in culture. Therefore, the residual chlorine present in water, if present, that can be neutralized by adding sodium thiosulfate. Then how to collect the water? There are various sampling points in the hospital premises, such as portable water samples from the various pipelines, endoscopy rinse water, dialysis water, operation theatre water. All these are various sampling points from where the water specimen may be collected. Okay, so apart from the water specimen, you can also collect swabs from the tap. So you can see that this is how you collect the water specimen from the tap. Okay, you have to wear gloves so that it will prevent the cross contamination of hand flora organism with the water. So you have to wear the pair of gloves and you have to collect the water. Apart from water collection, you can also collect the swabs. Okay, swabs from the tap may be collected. So the cotton swabs can be inserted into the nostrils of the tap, and it may be rotated thoroughly inside the pipeline, and from there you have to you can collect the swab. Okay, suppose apart from the hospital supply, various other community places, how to collect the specimen? They say that stream, lake, or swimming pool are the various community places from where the water sample may be collected. And here what you have to do is you have to insert a bottle. You have to immerse a bottle into the stream, lake, or swimming pool, and the bottle should only be opened only after immersing at least a depth of 30 centimeter, and at that time you have to open the bottle facing the current. The mouth should face the mouth of the bottle should face the current of the water. That time you have to open the bottle. Okay, you should not open the cap of the bottle then dip the bottle inside. You should dip the bottle along with the cap, and 30 centimeter depth you have to go down, then you have to open the cap of the bottle, and at that time the position of the bottle will be such that the mouth should face the current of the water. So this is how the water sample has to be collected. Now we will discuss the most common method, that is multiple tube method. Multiple tube method, as I told you, it is the most common method for water surveillance. It is widely used by the hospital as well as by the community water sampling, and it involves why it is called as multiple tube method, because it involves mixing a specific volume of water sample. A specific volume of water sample what is collected, that is been mixed in multiple tubes containing the medium. Here the culture medium of choice is McCrady purple broth. Okay, McCrady purple broth, why it is called as like that? Because it is purple in color. The broth appear purple in color. So the McCrady purple broth is the medium, is the culture medium what is used, and that is mixed with fixed volume of water in various tubes. For example, if you want to test for unpolluted or unturbid water, then the method is like that. You have to use 50 ml of water you have to add. So this is a 50 ml of water you have to add with one tube containing 50 ml of the medium. With one tube containing 50 ml of the medium plus 10 ml of water you have to add in 5 different tubes, each tube containing 10 ml of the medium. So all these are the 10 ml tubes. This is a 50 ml container. So all this will have McCrady purple broth, and McCrady purple broth appear purple in color. So what you do is in the 10 ml tube, what you do is you add in all these 5 tubes, you add 10 ml of water which need to be tested plus 10 ml of the culture medium, that is McCrady purple broth. And in the bigger tube you add 50 ml of water with 50 ml of the culture medium. Okay, so it should appear purple. Now it is looking yellow because the organism is grown. After the organism is grown, it will appear yellow. Otherwise, primarily when you add a water sample to the culture medium, it should appear purple in color. Okay, so the result is like this. If organism is not grown, if there is no organism in the water sample, the test will be negative. In such case, the media will remain as purple. Or if the test is a positive, organism is grown, then the media may turn to yellow color. So that is how you have to take the reading. After you take the reading, what you need to do is you have to determine something called as the most probable number. Most probable number means what? It is a number of tube giving the positive reaction is compared with the McCrady statistical table to determine the most probable number of bacteria present in water sample per 100 ml of water. That means what? There is a statistical table available called as McCrady's statistical table, and you have to compare how many bottles have become yellow color. That many you have to put into the table to see that what is the most probable number. So the most probable number per 100 ml of water sample is also called as presumptive coliform count. It is also called as presumptive coliform count. It indicates that there is a coliform organism contamination with the water. Okay. Now I will show you the McCrady's table. So you can see that this is the McCrady's statistical table. I will show you one example also. The same example what we have discussed. You have tested the water sample, and out of the 5 10 ml tube, 2 10 ml tube has turned yellow color, and 1 50 ml tube has turned yellow color, and 3 10 ml tubes they remain negative. So we will see what is the most probable number. So you can see here, among the 50 ml, okay, only 150 ml has turned positive, and 2 or 10 ml has turned positive. So this is the row which is applicable for us. So one 50 ml tube has turned positive, 2 tubes of 10 ml tube has turned positive. Therefore, the most probable number per 100 ml of drinking water is 6. It indicates that 6 coliforms are present per 100 ml of water. This is called as the most probable number of bacteria which may be present per 100 ml of water, and this will give you the presumptive coliform count. Why it is called as presumptive coliform count is because it is not confirmatory. You have to do a subculture to identify what organism it is. Overall, it will give you a preliminary clue that it is presumptive coliform organisms are present. Okay, so now how to know that whether the drinking water is of good quality or not? You have seen that you can count that what is the most probable number, but that most probable number of organisms which are present in the drinking water is significant or not, how you will know? There is another important table where you can classify the quality of drinking water supply by the most probable number of organisms which are present. So you can see here, if you have coliform count other than E. coli, if the count is 0, then it is taken as excellent. Count is 1 to 3 is taken as satisfactory. The count is 4 to 9, it is intermediate, and more than 10 is the water is said to be unsatisfactory. However, if the organism present in the water is a thermotolerant E. coli, then even a count of 1 per 100 ml is also called as unsatisfactory. In our example, we have seen that the most probable number for us is 6 organisms per 100 ml. So 6 organisms per 100 ml of drinking water. If you further process the organism to identify what organism it is, if it is a thermotolerant E. coli, then more than 1 is unsatisfactory. Therefore, 6 is taken as unsatisfactory. Suppose the identification of the coliform will become Klebsiella. If you have Klebsiella, then a count of 6 will be regarded as intermediate quality of water. Okay, so what is more important is, after you know it is presentable, coliforms are there, the next step is you have to confirm what is the identification. You have to make sure that whether thermotolerant E. coli is present or not, and that is done by a test called as Ekman test. Ekman tests are also called as differential coliform count. In this method, it is a continuation of the multiple tube method. First, in the multiple tube method, you will have yellow coloration of the McCrady purple broth. Then you will know that the presentive coliform organisms are present. You can also calculate the most probable number. Then you have to go for identification of the organism. Identification of the organism to know that it is thermotolerant E. coli or not, that is done by a method called as Ekman test or differential coliform count. It is done to confirm that the coliform bacilli which are detected, they are fecal E. coli or thermotolerant E. coli or not. How it is done? It is done by two methods. One is subculturing the positive tubes, the yellow color McCrady purple tubes. You can do a subculture on a lactose containing a medium. The classical medium what we use is called as brilliant green bile broth. So this is a brilliant green bile broth. It is a green color medium. It contains lactose. So if thermotolerant E. coli is present, thermotolerant E. coli are also called as fecal E. coli. It will ferment the lactose, and lactose fermentation will turn the medium into blue color. So you can see that lactose fermentation will change the color from green, and also the production of acid and gas production you will come to know by putting a Durham's tube, and you have to do the entire testing at 44 degrees centigrade, because thermotolerant E. coli, you have to know that the Escherichia coli is thermotolerant or not. That property can be detected by incubating the brilliant green bile broth at 44 degree centigrade. Okay, so a demonstration of positive indole test at 44 degree centigrade is another method by which you can identify the causative agent is fecal E. coli or not. Okay, so this is how the thermotolerant or fecal E. coli is detected. As you know that multiple tube method is the most commonly performed method, and it is the best method to perform for turbid water. Whereas if you have a clean water, if you have a dialysis water and various other hospital water supplies where the organism load is expected to be low, then the best method what can be followed is membrane filtration method. Okay, so membrane filtration method, the method here is the known volume of water, like for example 100 ml of water, you allow to filtered through the cellulose membrane filter of pore size. Various pore size can be used. 0.2 micron is the bacterial filter you can use, or even 0.45 micron also you can use. So you can see that these are the cellulose filters. These filters are placed at the base of the jar. This is a membrane filtration apparatus. It has a water filling jar and a motor. So, and in between the base is there where you have to place the filter. Okay, after you place the filter, you pour the 100 ml of water, and after you add water, you on the motor system so that the water will be sucked inside. The water will be discarded and will be sucked inside. What will be retained in the membrane is over the membrane the organisms may be retained. Okay, so after that what you can do is the bacteria which are retained on the surface of the membrane filter, so that membrane filter containing the bacteria can be transferred into a petri dish containing a suitable media. The most appropriate media what is used for membrane filtration is membrane lauryl sulfate broth. Okay, So you can see that this is a petri dish containing a membrane lauryl sulfate broth, over which you have to place the filter membrane. Okay, so I'll just show you again. This is the membrane filter. This is placed here in the base of the water containing jar. Then here you pour the water, then you switch on the motor, then the organisms, the bacteria will be retained over its surface. Then such membrane will be placed over a petri dish containing a medium called as membrane lauryl sulfate broth, and you incubate this for 24 to 48 hours. After this, what will happen is, if organisms are retained over the membrane surface, they will form yellow colonies. So you can see that various places the yellow colonies are formed. The yellow colonies will indicate the organisms are present, and please remember that these yellow colonies are characteristic that the coliform organisms are present. It cannot say that it is thermotolerant E. coli. For that you have to perform the H-command test, what we discussed for multiple tube method. But however, it will say that the coliform organisms are present, and you can count also how many number of colonies are present, and that many number of organisms are present in 100 ml of water. So membrane filtration method is a quantitative test where you can exactly count how many number of organisms are present in 100 ml of water. If you take initially 100 ml of water to perform the test, then how many colonies are grown, that will be your organism load present in 100 ml of water. Okay, confirmation. How will you know that it is thermotolerant E. coli? You by the membrane filtration, you will only know that the coliform organisms are present. Thermotolerant E. coli, you have to confirm by saying what we have discussed. You have to check for lactose fermentation and acid and gas production at 44 degree centigrade. The same what we have discussed for Eichmann test, a similar kind of test you have to perform here to check that at 44 degree centigrade it is fermenting lactose or not, along with gas and acid production is happening or not from lactose, and it is producing indole or not at 44 degrees centigrade. If these tests are positive, then you can say that it is thermotolerant E. coli. What is the advantage of membrane filtration method? I told you that it is the best method where the organism load is low. When the bacterial count is expected to be low, especially dialysis water, it is the best method to perform the water surveillance. And also when you have a large volume of water to be tested, membrane filtration method is the best method. It has got some disadvantage. The most important disadvantage is it is not suitable for turbid water. For turbid water, multiple tube method alone is important. The membrane filtration method is not suitable for turbid water, and it is more expensive than multiple tube method. Therefore, multiple tube method is by and large the most commonly used method. The membrane filtration method is used only for a limited setting such as dialysis water testing. Now the third method which is called as presence absence method. It is not a commonly used method. It is a qualitative method which will just detect that the organism is present or not, and various examples are there. One of the example is H2S coated strips can be used. The method is called as Manja's method, where H2S coated strips are used, where it can detect that the H2S producing organisms such as salmonella are present in water or not. So it is not a very reliable method, because it will not say that it is salmonella. It will only say that H2S producing organisms are present in water supply or not. So this is the third method which can be used. So now we will see how to test for water contaminated with healthcare associated pathogens. So I told you that there are two categories of pathogens in hospital. Drinking water may have enteric pathogen, and other than that, the hospital water supply may have various hospital multi-drug resistant organisms, or maybe the unique organisms which are found in the hospital water bodies such as Legionella. So how to detect those organisms? So here the methodology is the most hospital acquired pathogens are recovered by membrane filtration method. Okay, so membrane filtration method is the method of choice of testing of water supply in hospital. After that, you have to plate in suitable culture medium. For example, if you want to recover Legionella species, then the culture medium what is used is called as buffered charcoal yeast extract. Okay, buffer charcoal yeast extract or BCYE medium is the recommended medium for the recovery of Legionella. So similarly, what organism you are suspecting, the respective suitable culture medium is used after the membrane filtration is over. Otherwise, you can also use the lauryl sulfate medium what we have discussed for drinking water supply. The same lauryl sulfate medium you can also use for detection of various multi-drug resistant bugs in the hospital water supply. Another important thing which the hospital water may get contaminated is with bacterial endotoxins. Endotoxin means what? Endotoxins are the component of gram-negative bacterial cell wall. So they say that the dialysis water and the devices used for dialysis, they may get contaminated with the endotoxin, and in such case, if you take such endotoxin contaminated water for dialysis, then this endotoxin inside the human body may induce various inflammatory responses, and therefore it can cause various toxic side effects. So dialysis water should be devoid of bacterial endotoxin. Okay, therefore the dialysis water should be tested for presence of endotoxin. The dialysis water which is used for hemodialysis purposes should be regularly tested for the presence of endotoxin. And what is the permissible level? They say that the water used to prepare the dialysis and to reprocess the hemodialysis should contain an endotoxin level less than 0.25 endotoxin unit per ml. So this is a cutoff which has to be followed. So below this cutoff, endotoxin, if it is present, it is safe. It cannot induce inflammatory response. But if the endotoxins are present above this cutoff, it is risky to use those dialysis water, because that can induce inflammatory response inside the human body. Okay, there are various methods available to detect endotoxin. The most common method what is employed by the hospital is gel clot assay or limulus amoebicide lysate assay. It is called as LAL assay, or limulus amoebicide lysate assay is an example of gel clot assay. This is the most recommended, the most popular method to which is used by hospital for the detection of endotoxin. Okay, so this is all about the water surveillance. Now we will see some MCQs. Which is not used as indicator organism for fecal contamination of water. Please remember that Vibrio cholera is an enteric pathogen. Enteric pathogens are present in very minute quantity, therefore they cannot be directly detected in drinking water. You have to use various indicator organisms for detection of fecal contamination of water. Among this, the most reliable indicator is thermotolerant E. coli, also called as fecal E. coli. The next question for you is, indication of water surveillance include all except. Please remember, routine periodic testing of hospital water supply is strictly prohibited. You should not just periodically test the hospital water supply. You have to do the water surveillance only when there is specific indications such as outbreak investigation, for research purposes, or for the quality assurance to evaluate that the new infection control practices which are introduced are working properly or not. So these are the indications for water surveillance. Routine periodic testing of hospital water supply is not indicated. The next question for you is, membrane filtration method has the following advantages from multiple tube method except, which is not an advantage of membrane filtration method. Remember that turbid water cannot be tested by membrane filtration method. The most reliable method for testing the water surveillance of turbid water is multiple tube method, whereas others are the advantage of membrane filtration method. So this is all about the water surveillance. So what are the indications of air surveillance? Remember that like water surveillance, even air surveillance also should be performed only when specific indications are present. Periodic routine air surveillance is not indicated. Okay, so the specific indications include investigation of an outbreak for research purposes, after reconstruction or renovation of the operation theater. In all those renovated situations, you have to test for air quality. And after fogging, suppose you are doing fogging of the operation theater, to ensure that whether the fogging has been done appropriately or not, also you can perform air surveillance. But for short term evaluation of a change in infection control practices, suppose you have introduced a new disinfectant or you have introduced any new infection control practices, then to know that it is working well or not, you have to perform air surveillance. So the indication of air surveillance is more or less same as indication of water surveillance. Please remember that the periodic routine air surveillance is not indicated. Now we will see what are the various methods available for air surveillance. Air microbiology can be tested by various methods. So you know that air surveillance is most commonly performed in operation theatre. So what are the various methods available to evaluate the air quality in the operation theatre? The methods can be classified into microbiological method as well as non-microbiological method. Microbiological method like passive monitoring by a settle plate method, active monitoring by slit slampler method, and air particle counter method. So let us discuss one by one. The first method is passive monitoring, or also called as settle plate method. In this method, what we do is the various culture plate, for example blood agar, are exposed to the air of the operation room for a given period of time, and it has been incubated for 37 degrees centigrade for 24 hours. So the most common method what has been followed is called as 111 method. In 111 method, the blood agar plate is placed at different locations of operation theatre, 1 meter away from the side wall, 1 meter above from the floor, and for a duration of 1 hour. Okay, so it is a method where the blood agar plate is just kept in various location of the operation theatre. And why it is called a settle plate method? Because you are allowing the microorganism to settle down over the culture plate, and it is a passive method because you are not actively sucking the air, you are just placing the culture plate in the various location of the operation theatre, and you are allowing the organism to passively come and settle down over the culture plate. And where you have to place the culture plate in the operation theatre, for that 111 method is used. That means you have to place the culture plate 1 meter from the side wall of operation theatre, 1 meter above the floor level, and for a duration of 1 hour. Okay, so after that you have to incubate this at 37 degrees centigrade for 24 hours. So you can see that wherever the bacteria would have settled down, the colonies may be formed at the respective places over the blood agar plate where the bacteria will be sedimented or settled down. Okay, so the number of colonies will give an indirect estimation that how many organisms are present in the air of the operation theatre. Now we will see what are the acceptable levels. So you will count how many colonies are there in the culture plate, but how much of organism load is acceptable, for that various criteria are used. One of the criteria, they say that if the organism load is less than 5, colonies in a 9 centimeter diameter plate, that is equivalent to less than 786 colony forming unit per meter square per hour. So this is the acceptable limit at rest. And when the operation theater is been operational, when the OT is been operational, the count will increase. So for that, the acceptable count is less than 25 colonies in a 9 centimeter diameter blood agar plate. So usually the blood agar plate what are available will be of 9 to 10 centimeter diameter. So in such a blood agar plate, less than 25 colonies while the OT is operational, or less than 5 colonies while the OT is in rest per hour is considered as acceptable. Above this cutoff, it is considered as unacceptable. You have to do environmental cleaning of the operation theater. Now we will see the various disadvantages of membrane filtration method. Membrane filtration method is not reliable for smaller particles that are suspended in air because they will not settle down over the culture plate. It cannot quantify the volume of air sampled. Okay, for that you have to do active sampling method. Passive sampling method cannot reliably quantify how much of organisms are present in air. And also for fungal spore detection, this is not a reliable method because the fungal spores can suspend in the air for a longer period of time. They will not settle down over the plate so easily. Now we'll see the second method. The second method is called as active monitoring or slit sampler method. In this method, we use a equipment called as air sampler or sieve impactor. So sieve impactor is an example of air sampler. So air samplers are used. So you can see this is the equipment which is called as air sampler. This equipment has a vacuum pump. Okay, so this is the vacuum pump of the equipment, and it has a perforated lead. It has a perforated lead. The lead has got various perforations. So what you do is you have to open the lead. Okay, in this equipment you have to open the lead and you have to place a culture medium here. After that you have to close the lid. I repeat, in this air sampler the first step is open the lid. The lid will have perforation, so that lid you have to open completely. It will be screw capitally tight, so you can unscrew the lid and you can open it from the equipment. After that, a blood agar plate you have to place over the equipment, then you have to close the lid. After that, what you have to do is you have to on the machine. When you on the machine, the vacuum pump will work, and the vacuum pump will physically draw a known volume of air through the perforated lead, and it allows it to fall over the agar plate. It allows it to impact over the agar plate, and therefore it is called a sieve impactor, because the perforated lead will have holes or sieves, and through which the air is sucked inside, and it is allowed to fall over the culture plate. It is allowed to impact over the culture plate, and at the point of impaction you will have colony formation. The colonies are formed at the point of impaction, provided the organism is present in the air. If there is no organism in the air, then no colonies are formed. Okay, so following the incubation, after this process is over, you need to incubate the plate, and following the incubation, the quantity of the microorganism present in the culture plate is measured in terms of colony forming unit per meter cube of air. Okay, so that is how you have to quantitate the microorganism. So the advantage of air sampler method is it can reliably quantitate the organisms which are present in the air. Okay, so it is useful for operation theater, bone marrow transplant unit. All these are the various location where the air sampling can be done by the air sampler or sieve impactor method. Okay, Now we will see the third method. The third method is called as air particle counter method. The air particle counter method, the various equipment are available which are called as air particle counters. They will reliably count the airborne particle, how much of airborne particle which are present in the hospital air, in the operation theater air. In the real time they can quantitate. Okay, so they will follow a laser technology which will generate a fluorescent signal from the viable air particle. That is how you can quantitate the number of air particles. After the particle counts are obtained, you have to match that the particle counts are significant or non-significant. So the particle counts, the air particle count of an operation theater is considered acceptable. Okay, it is considered acceptable only when it falls in the acceptable range of a clean room standard as laid down by ISO. ISO is International Organization for Standardization. They have given various clean room standard, and it should match with the clean room standard of ISO. Okay, for example, most of the HEPA filtered operation theater, most of the operation theater, the general principle is it should satisfy ISO standard level 6, and the level 6 clean room standard indicates that the room should have less than 35,200 particles of size 0.5 micron or less. Okay, I repeat, ISO level 6 indicates that the room should have less than 35,200 airborne particles of size less than 0.5 micrometer per mmq of volume of air sampled. So that is how the ISO 6 standard is there. So ISO 6 standard should be satisfied by most of the HEPA filtered OT. There are some specialized OT that is called as ultra clean OT. The example is very orthopedics OT. They should have even more stringent condition. They should satisfy ISO standard 5, which indicates that the room should maintain less than 3,520 particles of size 0.5 micron per mmq of volume sampled. So air particle counter are the most reliable method for determination of air microbiology. So there are three methods. First is settle plate method, then is air sampler method, and the most reliable method is air particle counter. All these are microbiological methods. There are various non-microbiological methods are also available. The non-microbiological method include the various standards like air velocity, humidity of the air. All this you have to regularly monitor in the operation theater. For example, air exchanges per hour in the operation theater, the guideline says that there should be at least 20 number of air exchanges per hour, out of which at least 4 air should be fresh in nature. So minimum 20 air exchanges per hour should take place in operation theater. So the next is air velocity. They say that 25 to 35 feet per minute is the air velocity which has to be maintained, and it is checked by a instrument called as anemometer, which will check air velocity. And the airflow direction should always be unidirectional in the operation theater, and it is downward, it is unidirectional and downward from the operation theater table. Okay, so that is how the airflow direction has to be. Next is the positive pressure. They say that the operating room should have more pressure than that of outside environment. Okay, the positive pressure between the OT and the adjoining area should always be there. It is required to prevent the outside entry of air into the operation theater. If inside the operation theater, if the pressure is more, then it will prevent the entry of air from outside environment into the operation theater environment. And the minimum pressure which has to be maintained, they say that the positive pressure differential between the operation theater and the outside environment has to be at least 25 Pascal or more. This is a recommended positive pressure differential which need to be maintained between the operation theater and the outside environment. Then coming to operation theater temperature, they say that 21 degree has to be maintained in general in operation theater, except for orthopedics OT where 18 degree centigrade need to be maintained. Relative humidity is around 20 to 60% which need to be maintained. Then coming to air handling unit in the operation theater, they say that there should be a dedicated air handling unit in the operation theater, which is specific for each operation theater. You should have a dedicated air handling unit. Air handling unit is a unit which will provide the low temperature air conditioning temperature will be maintained by the air handling unit, and also it will provide the fresh air into the operation theater. So just like that, the window AC or split AC you should not use. You should use always a dedicated air handling unit to provide the adequate temperature to the operating room. Okay, the reason why the window and split AC you should not use is these window and split AC are pure recirculating unit. So they only recirculate the operation theater air again and again. Therefore, they cannot take out the air to the outside environment. They cannot provide fresh air into the operation theater. Therefore, you should have a specialized dedicated air handling unit which not only will maintain the low temperature in the operation theater, but also will remove the air to the outside and will provide the fresh air every time. They say that at least 20 air exchanges per hour should happen. 20 air exchanges, okay, per hour should happen, and that will be performed by a dedicated air handling unit inside the operation theater. Okay, so they say that in the air handling unit, what it performs is the air it supplies inside the operation theater after passing through a terminal HEPA filter which is present in the ceiling of the air handling unit. Then the filtration area also, they say that it should extend 1 feet on each side of the operation theater table to cover the entire OT table as well as the surgical team, because the surgical team also will stand next to the OT table. So if you have if this is the operation theater, this is the OT table, then the OT table as well as little surrounding of the OT table, 1 feet area surrounding the OT table, this is the area which the air coming to this area must be filtered. Okay, so that filtration is done by use of a terminal HEPA filter. Okay, the next property is how much of standard need to be maintained. This we have already discussed. The general OT should maintain ISO class 6, whereas specialized OT such as orthopedic OT and all that, they should maintain ISO class 5. Okay, now we will see the wall, floor and ceiling. They say that the wall, floor and ceiling should be of anti-static type and made up of non-porous, smooth and seamless material. Okay, the floor, wall as well as ceiling should be made of non-porous, smooth and seamless material inside the operation theater, and the paint which is used for operation theater, it should have antibacterial and antifungal property. And next is how many people should occupy in the operation theater. They say that at any given of time there should not be more than 5 to 8% inside the operation theater. Okay, so you should have restriction of people, because more are the people inside the operation theater, there is more chance of contamination of hospital air inside the operation theater. Okay, so these are the various non-microbiological parameters which need to be maintained inside the operation theater. So this is all about air surveillance. Now we will discuss surface surveillance. Surface surveillance means what? The environmental sampling need to be performed from various surfaces of high risk area like operation theater. Okay, so it is why the surface surveillance is done. It is used to determine the source of environmental contamination. So if you have some surgical site infection outbreaks, SSI outbreak, if you have in a hospital, then you need to perform the surface a of course air surveillance also you have to do. Along with that, surface surveillance of the operation theater need to be performed. So what you do is, as I told you, high risk locations such as OTs and ICUs, the surface surveillance may be conducted, and the high touch areas of those locations need to be sampled. What is the indication of sampling? The surface surveillance is done usually for an outbreak investigation. The routine periodic surface surveillance is not indicated. Only for an outbreak situation you can do surface sampling. And how to do sampling? They say that you have to use moistened sterile swab. The sterile cotton swabs can be used, which can be soaked in a sterile saline, because the swabs need to be moistened, and such moistened swabs are used to collect the samples from. Please remember, you cannot collect from the entire operation theatre, so therefore the high risk areas where the chance of contamination is more, on all these high risk areas you have to collect the sampling, and after that you can inoculate onto a suitable media like blood agar. Okay, now you only the pathogenic organisms isolated are reported. The contaminant such as aerobic spore bearers or coagulase negative staphylococci, all these are not reported. Only the pathogenic organisms grown in surface sampling need to be reported. Okay, so this is all about surface surveillance. Now we will see some MCQ. Evaluation of quality of air in operation theatre can be performed by all method except remember that presence absence method is a method for water surveillance, not for air surveillance. These are the method for air quality surveillance. The next question for you is, quality of air in OT include all except. Again, a point to be noted, the minimum air exchanges per hour inside the operation theatre has to be 20. Okay, 20 air exchanges per hour has to be there. In that, 4 air should be fresh air. Okay, so this is a wrong statement here. Not a minimum 4, minimum 20 air exchanges you should have inside the operation theatre, and among which 4 should be fresh air. Other points are correct statement. You should have air velocity this much, airflow direction has to be unidirectional, and you should have a positive pressure maintained in the operation theatre, which has to be at least 2.5 Pascal. So this is the end of the session. We have discussed the various environmental surveillance methods in this session. We have discussed first the water surveillance, like the multiple tube method and membrane filtration method. Then we have discussed the air surveillance methods such as the settle plate method and the air sampler method. After that, we have discussed surface surveillance method. I hope you have understood the session. Thank you.